Aloha and mahalo for joining us for our HISAM webinar. HISAM stands for Hawaiʻi Invasive Species Awareness Month. It's an annual event dedicated to educating and mobilizing residents of Hawaii to protect our islands from the threats posed by invasive species. This year, we celebrate and reinforce our collective responsibility with the theme, Who protects Hawaii? You do. Invasive species are everyone's kuleana. And for more information on that, we'll be dropping a link in the chat as well. Today's talk, as I've mentioned, is Safeguarding Hawaii's ʻōhia trees through the ʻōhia Disease Resistance Program and the Myrtle Rust Monitoring Network. Our speaker is Dr. Mark Hughes. More about Mark in a minute. I am Kim Rogers, an outreach specialist with Kauai Invasive Species Committee, also known as KISC. And also I want to thank those two people who are kind of running the things behind the scenes. We have Kylie Lefebvre and Elizabeth Spieth. So today's speaker is Dr. Mark Hughes. Mark Hughes is a native of South Florida and earned his PhD in plant pathology from the University of Florida. His research focused on a lethal disease affecting avocado and bay trees known as laurel wilt. After completing his studies, he made a move to Hawaii Island in 2016, where he joined the plant pathology lab led by Dr. Lisa Keith. His work there centered on investigating the biology and management strategies related to rapid ʻōhia death, also known as ROD. Currently, Mark serves as a research plant pathologist as the U.S. Forest Service Institute of Pacific Islands Forestry in Hilo. His ongoing research not only delves into the intricacies of ROD, but also addresses other tree diseases that have negative impact on Hawaii and the Pacific Islands. Welcome, Mark, and thank you so much for agreeing to share your research and knowledge with us today during this month.
Thank you, Kim. I'm glad to be here. And thank you, everybody that's joining, that took the time out of your day to watch this presentation. I'm going to take a second and put up my presentation. Give me 1 sec here. Share screen. Can you see my screen, Kim, at the moment?
No, not yet.
Okay. Okay, there we go. I just had to pick the window.
It's a multi-step process.
Yeah, and then I'll get into the slideshow from the beginning, and I'll hide this bar at the bottom. Does that look okay, Kim?
I'm not seeing the slideshow yet. I'm seeing your working document.
Let me go back then. Let's see. It's a little... Let me pick a new window, I guess. Hide. From beginning. And you're not seeing that, are you?
I'm seeing you clicking around on that. So I'm seeing, like I said, kind of your working document that has the slides numbered on the left.
Okay, give me 1 sec. I'm going to play around and you can just I'm going to stop sharing and let me try this again.
Yeah, sometimes it helps to start over.
I think what it is I have multiple monitors, and when I turn them off, everything starts acting up on me. Share screen. Window. Entire screen. Let's try that.
Of course, it worked fine. Slide numbered on the left.
Yeah, okay. I'm sharing my entire screen, so hopefully now? You see it now, Kim? Interesting. I can't hear.
Yes.
Okay.
Now sorry, I was on mute. We are good to
go. Okay, cool. Thank you. Sorry about that, everyone. This worked fine like 2 minutes ago, but here we are. Yeah, so today I'm going to be talking about some research that I've been participating in, looking at work of our ʻōhia disease resistance program, and then another study or program looking at monitoring for a different disease called Myrtle Rust. So, for those that are not familiar with ʻōhia, it's a Metrosideros polymorpha. This is it's a hardwood tree endemic to Hawaii, and it's the dominant native tree in forests. It's been estimated to be about 80% of the biomass in the native forest, about 50% of all the forests in the state. What's really important about it is it's this pioneer species that can colonize these lava flows, as you can see on the photo here. This is a small ʻōhia plant growing at Hawaii's Volcano National Park. You can see how seemingly inhospitable this environment would be for plant growth, but ʻōhia really is very important because it can start colonizing that land. And over time, areas like this could eventually become forests. So it's really important for the turnover and the change of land in the Hawaiian Islands. And because this is such a keystone species, it's known to support a variety of plants and animals. It's been estimated about 500 arthropod species are associated with ʻōhia, and 18 of the 19 threatened and endangered bird species also have an association with ʻōhia. So again, very key to native forests. Like the species name says, polymorpha, it has many forms. It can exist on a very short stature tree or short stature plant to something that is a large canopy tree, meters in diameter. So it has a lot of variability. And that is really good when we think of disease resistance, because we want to have a lot of variability in the forest, because that is the bank of basically where we're looking for these genes that can potentially confer resistance to rod. And these are some facts I pulled up giving a talk on the mainland, really just trying to, it's been said a lot that ʻōhia is really vital to the watershed. So I wanted to really figure out, like, what is exactly does that mean? And through some literature searching, it was found that, it's not even just the ʻōhia itself. It's the leaves, the ferns, the moss that are epiphytic on the plant are really good at capturing mist and humidity from the air, even without rain. There's been studies done showing that compared to, let's say, the non-native strawberry guava, ʻōhia forests can capture about 50% more moisture, and which is really good for the aquifers and the streams, because that is the water that we rely on for drinking, agriculture. It's what supports life here on Hawaii. So it's very important. And also it shows that it's been shown that ʻōhia forests store a lot of carbon compared to other forest types across the island. So again, just showing into that whole role of how important it is as a host or a species. And I think it's boiled down really nicely in our ROD strategic plan from 20 to 2024, where it was written that the native Hawaiian forest is essentially and fundamentally an ʻōhia forest. And this is showing kind of a nice, young, healthy ʻōhia forest that is free of disease. And this is another photo from JB Friday in the lowland puna forest. This is before rod. This is really what you could see out on a hike. And then unfortunately, once the disease takes a hold and starts infecting and killing trees, you can get this dramatic change in the forest, in its composition, the massive amount of mortality that occurs. So it's really a game changer in regards to Hawaiian forests. This is kind of another overhead view of showing the different, how it would look at different stages of decline and death. So what we're hoping to do through the ʻōhia disease resistance program is to identify trees that can resist the rod pathogen, in particular Ceratocystis leucohea, for restoration of native landscapes, and also for the perpetuation of ʻōhia in our biocultural landscape. This project was really started in 2018 by a graduate student named Blaine Louise. He was working out of Lisa Keith's lab at the USDA ARS in Hilo, right next to the Forest Service. And this was his master's thesis project, really looking to see if there was variability between different ʻōhia lineages, different varieties. And that was really kind of the start of this type of work that evolved since then. Now, this is more than just um, a closed kind of group. There's a lot of people involved in this work, from um, the Forest Service and the Akaka Foundation, which are kind of the core of that group, but also ARS, University of Hawaii, DLNR, DOFA, and a lot of other organizations that have contributed in one way or another. So it's definitely a group effort. And lastly, I like to say that, the majority of the financial support has been provided by the Forest Service and the state of Hawaii DLNR, DOFA. So we're very grateful for continued funding that keeps this project afloat and moving forward, and really help giving us the time to make progress. So this is when I say ODRP, that means the ʻŌhiʻa Disease Resistance Program. And it's based out of the Forest Service Institute of Pacific Islands Forestry in Hilo, Hawaii. And it was originally, the program itself was kind of started by Christian Giardina, incorporating Blaine's project into it. That was the first actual screening that was done. He's now the director of IPIF at the Forest Service, and then Kainana Francesco, his technician. Since then, I've kind of moved into the role of the lead advisor for this work, and we've hired on staff to really help support it. And these are the people that keep the project going on a day-to-day basis. So without them, there really would be no ʻŌhiʻa Disease Resistance Program. So we have Chloe Martins, Kali'i Homalo, and her sister Chantilly. They work in the greenhouses and various other community outreach portions of the project. Ryan Belcher is our coordinator, and he's kind of the manager of the program on the day-to-day basis. And then Nainoa Gu helps with the greenhouse and the field coordination and implementation of the project. So this is the team, the kind of like the core team on the day-to-day. And this is run out of our nursery here at the Forest Service. You can see some benches behind us where we're growing ʻŌhiʻa. So if you'd like information on just the background of this project and program, you can look up in the journal called New Forest. It's an open access journal. If you type in rapid ʻŌhiʻa death, you can get this framework document that was really trying to consolidate and get our ideas crystallized on paper to show what our goals are for this project and our strategies of how to implement them. So it's kind of a background information if you're interested. So now when we're talking about rapid ʻŌhiʻa death and how to screen for resistance, the first thing we have to look at is how are we going to collect the material used for this disease resistance screening work. And there are two strategies that we are taking. The first one I'll talk about is the use of survivor trees in areas of intense levels of mortality here on Hawaii Island. So what's good about this is when we look at survivor trees, we know kind of what isn't resistant because those have already been unfortunately killed off by the disease. So now there's this remnant population of trees. And the idea is, well, why are they there? Is there something special about them? Did they randomly just escape infection, or were they actually inoculated or infected, but they have some level of genetic resistance that's able to fight off disease? And with that work, what we do is we take rooted cuttings. So we'll go to the survivor trees, we'll take small branch cuttings, and then we have a sealer that we can seal up the wounds. And when I refer to those cuttings, I'm referring to those as clones because they're clonally propagated. They have the exact same genetic background as the mother tree in nature, and it allows us to do a lot of testing on those genetics without actually harming the tree in the field. And what's good for this. It's really nice because for screening, the genetics are very constrained, but and it's good for small areas. It's not great for large scale restoration because, again, you don't want to be replanting mountainsides with just a few clones. So it has its role, but it's really what helped us get started in this program. And then what we're kind of moving over to now is using seed collections. So we'll collect them from our collaboration with the seed banks in Hawaii, or we'll do new collections from areas. Specifically, we're focusing from areas with a lot of rod mortality. And these trials started in 2023. And this is really traditional resistance screening, traditional breeding, where you're getting what is called a seed family. So if you go to an individual tree in nature and you pull off a flower or a seed pod, which forms after the flower, you're going to have hundreds to thousands of seeds from that individual tree. And that's what we call a seed family. So they're all related, but they're not clones of each other, because the pollen that comes in, these are what we call open pollinated. So the pollen that comes in is just coming from the environment. So the maternal genetics are the same, but the paternal genetics, the pollen is different. So you're going to get more variability between the seed families, but that's really good when you're talking about restoration, because you want a lot of genetic variability in these populations for outplanting. So kind of going back to the clonal work, talking about these survivor ʻōhia trees, this is a picture at Steinbeck Highway in Hilo. And you can see all the dead ʻōhia that have passed over time since infection started here. This is one of those early hotspots, a lot of intense mortality. We've used it for a lot of studies. But what you can see is we have this single lone survivor tree that is persisting. And it could be one thought is it's an escape. It just didn't get inoculated. But again, if you look around, all the other trees have been inoculated and died. So it raises the question of, is there really some resistance in this tree? So this was formed, we did this under a study, a Forest Service grant that funded this study. And we were looking at resistance in these survivor trees, but we were also looking at differences in forest stands as a whole. So we went to these high mortality sites, but we also looked at adjacent forest stands that had little mortality. And we were trying to figure out, do you have to go to high mortality sites to get resistance? Or is there really no association? Does site not matter? Is it more random? Can we go to these low mortality sites and also have similar levels of resistance? And that's something we're still looking at through statistical analysis. What's also interesting is that what's kind of fun is that this has been aided by remote sensing. We've been able to use satellite imagery to help us pick out trees, which is really nice. It makes things faster and streamlines our selection process. So we have 4 sites selected on Hawaiʻi Island. IPIF, which is around the Forest Service property, KMR, which is Keukaha Military Reservation, on Steinbeck Highway, which was this area you just saw, and then Pu'ukaliu, which is in Kamehameha School's land by Leilani Estates, really kind of where it was first detected and a lot of mortality. So we have about 200 trees that were selected for propagation. We grow the rooted cuttings in a mist chamber, and we grow them and then seed. If we collect seed, if it's available, we sow those in small dibble tubes, and we grow those plants until they're about 6 millimeters in diameter. We need a minimum stem diameter to do the inoculation. So 6 millimeters is between kind of a pen and a Sharpie in diameter. And it takes about 18 to 24 months to get them to that size. And Ryan and I know are trying to speed up that process, where we've kind of changed up some of the pots that we use. And I think the goal is to get there faster so that we can, screen faster, because already you're talking about 2 years before we even start the screening trial. This is just showing kind of, we have an outdoor nursery. We grow all our plants outside, full sun, and overhead irrigation gets us nice, strong plants. And then we have kind of an indoor inoculation screening greenhouse. This is where our inoculations occur. This is all the plant material before the screening process. And so I'll talk about how we screen. So for the disease resistance work, we're really focusing on Ceratocystis leukou ʻōhia. This is the more virulent of the 2 pathogens. This is the one that is responsible for the majority of this widespread mortality that's expanding. So this pathogen is present on Hawaiʻi Island and Kauai. But again, it's the more aggressive, it's the more important of the 2. So we can grow the fungus on culture plates. And then what we will do is we will flood those plates with water. We're able to dislodge the spores. We can quantify it to a certain concentration, and then we will drop essentially 20,000 spores onto these pieces of filter paper. So this is just kind of a lab paper. We'll incubate them for 24 hours in the incubator. And what will happen is you can start to see there's this little fuzzy ring here. I don't know. Hopefully you can see my mouse. And that's germinating spores. So that lets us know that, okay, this inoculum is viable, it's living, it's growing, and that means it's good for inoculation. So the process is here showing the different the plants. We'll make an incision into the sapwood with a scalpel. So we want to get it direct. We want to make that wound so the fungus is directly interacting with the xylem tissue. We'll score it a little bit to make little micro wounds. So again, the spores can lodge in and nestle in. We'll add this filter paper disc, and then we will wrap it with a cotton ball. And that's really to keep that inoculation site moist to give more time for the fungus to start growing in the plant. So we're hedging everything to make sure that we have a good inoculation, high chances of infection, because again, we want to screen robustly and make sure we don't have things surviving just because they didn't get a good inoculation. We rate on a 0 to 5 scale based off of what you see externally, the leaves, the canopy. So it's in 25% increments, from 0 being asymptomatic or no wilt to 5 being dead. And then, yeah, like I said, 25% in between. And we do this about twice a week. We do ratings. This is some preliminary data, but I guess what I'm really trying to tell you is we're working with a statistician, a Forest Service statistician, Nels Johnson, and he's developing these models that, beyond just looking at the scores of resistance, we're trying to figure out, are there other factors associated with this. So we're looking at variety, forest site, again, that high versus low mortality. Does elevation play a role? Does substrate play a role? Does precipitation play a role? Because some of these factors, these environmental factors, can influence the morphology of in the vascular system of ʻŌhiʻa. And so we're kind of trying to get as much out of this study as we can in regards to data analysis. And Nels is developing those statistical models to look at it. So I'm going to present some data on the first trial. This was a 2021 trial. This is one that Blaine and I overlapped on. So to kind of guide you through this table, you have your disease severity score of 0 to 5. 0 is no symptoms, 5 is dead. And each bar represents replicates, clonal replicates of a particular tree out in the field in nature. So basically what you could see is we have a spectrum of responses. And that's really the main take home I'm trying to tell you with this particular study, because we're still in the process of doing the statistical analysis. And so I don't want to really focus on one tree versus another. The main take home is that we're seeing a good gradient of symptoms and levels of resistance. And that's really important because in some symptoms, in some systems in plant pathology, there's very little to no resistance that can be found. So even just having this result here showing that everything isn't dead is good. And not only is everything not dead, but we have certain genotypes that hold up very well. Of course, we have to repeat this and do this a few times to really get confidence in the levels of resistance. But it's really encouraging. The other thing that's encouraging is we get a lot of mortality, and that's showing that our inoculation system is pretty robust and it's able to kill trees or kill plants. So, both of those, having a lot of mortality and very little mortality, is good in my eyes, for screening. So this is a 2021 trial. We did there's 56 clones, so 56 mother trees have been represented here. And then this is another 2023 trial we did, again, with more clones. And you could see kind of the same pattern. We have a good chunk of mortality, which is fine because we know that resistance is probably, it's needle in a haystack, I guess you could say. It's probably not expressed throughout every tree. So again, this is a fine pattern. that we're seeing. And again, kind of towards this end, we arbitrarily are saying anything under a 1, anything under 25% wilt is kind of resistant. But all of this material has usefulness in studies and different things. So again, we're excited to see those levels of resistance. And then this is our 2023 trial. Now, this is actually looking at seed families. This is not clones anymore. These are, again, when we went to these trees to collect cuttings, if there was seed available, we would collect the seed, we would grow them out. And then this is really our first look at resistance within a seed family. So what's different here, again, is the seed family is going to have more variability between plants because it's open pollinated. But again, we are seeing, levels of resistance and variability. Again, all this is preliminary and needs to be the best ones need to be screened over again. So as we do these trials over and over, and these genotypes go through more screenings, our levels of confidence will build and their durability over the long term. So moving forward, what we have to do is we're going to confirm levels of resistance with follow-up trials. At Keaukaha Military Reservation, we have a really good working relationship with them. And they've cleared about 2.5 acres of strawberry, guava, and ironwood for us, as you can see here, put a fence around it, and all the material that has survived inoculation will be outplanted there to see how it survives in an area with a lot of rod. There's a lot of rod mortality in this area. But really, it's also for germplasm. We're trying to preserve this germplasm that's surviving and use it as a source of cuttings and seed for future work. So we don't have to go back to the mother tree over and over again. We have new representatives of those genetics here in our farm where we can maintain them and grow them to the size we want and fertilize them and kind of make it more like a farm. We've also started screening commercial ʻōhia clones, and I'll have results on that. We're doing it now, but these are things that are available in the landscape nursery trade. So we want to see if there's any resistance out there. And another thing is, we want to know, does resistance pass into seedlings? So if we had a clone that did well, does seed from the clone do well? And that would show you that this resistance could be kind of passed on the maternal side. And moving forward, the majority of our efforts now are going for the seed family, seed collections, because again, that's going to make the most impact in regards to outplanting. So we're expanding our geographies of seed collection, still focusing on rod impacted zones because we've been working with land managers that are basically asking us for resistant material. And we want partners that are ready to outplant because that's really the goal, is to have material that can be put back into the ground to get ʻōhia back on the landscape. And the next steps really depend on what we find. If we find these mother trees that produce seed and the whole family has really high levels of resistance, that kind of can mean one thing, where maybe we can tell the manager, oh, these trees that you have on your land, the seed they produce right now, as is open pollinated, is really resistant. And you can just go to those trees and pick seed. And that could be your starting genetics for outplanting. If there's low levels of resistance, then we might have to do what's called breeding, where we can grow these in an orchard and then do crosses and try to up the levels of natural resistance. The good thing is in talking with some breeders, generally the first round of crosses can give you a lot of genetic gain and resistance. So we'll just have to see what we get as we screen this material. And it's probably going to be variable site per site, family per family, but those are kind of some options we can do moving forward. Okay, Kim, this is my halfway point, at least in regards to subject matter. So I can leave it open for questions. If anybody has questions about this part, I'll go back to the rod.
I don't see any questions in our Q&A, but if anybody has any that wants to pop one in and or pop it in the chat, that's fine either. While you get a little drink of water and folks maybe think about their questions, I have one or two for you.
Sure.
I'm kind of curious because I get asked this when I do outreach. When you're rating the trees that 0 to 5, 0 healthy, perfectly healthy, 5 completely dead. Do you ever see any that go from in the resistance trials, let's say get to a 4 and then come back down to a 3 or a 2? Or is it just thinking that the resistance prevents the disease progression altogether?
Um, well, we sometimes we do see, it would go to, let's say, a 4 and then go down to a 3. And really what that is that's like resprouting at the base or lower on the
Okay.
tree.
Right.
And that's probably not going to last long term. That's usually maybe the tree had enough energy to put another kind of grow a little more xylem, and then put some sprouts out. But generally, that doesn't. Um, we don't think that persists, and that's not really what we want. We don't want a tree that basically dies and then put some sprouts back on it. We're looking for trees that don't really even get to that point. And the mechanism of resistance we don't know, but if we look at other systems that are similar, it's probably that the tree is actually restricting movement of the pathogen um, throughout. So it's probably there's a way that the xylem can occlude it and just keep the fungus kind of restricted to that inoculation site. That's what my guess is for the mechanism of resistance. And that's really what we're shooting for is
Okay.
low levels of infection overall.
Okay. That makes sense to me. Um, and let me just check. We do have a question that popped in from Kirsten. Are there plans for the selected resistant clones to be tested downstream for other pathogens like Neofusicoccum parvum, pardon me if I've mispronounced that, to create a multiple resistant strain?
Yeah, I mean that's also been asked of just Julio here, and there's no plans right now, just because it's a capacity issue. If we had endless funding and endless staffing, yeah, we could do that, but we're really focusing on rod and kind of the most virulent pathogen of rod. I do work on that other pathogen, the Neofusicoccum, and the canker that um, it causes, and that the pathology there is still a little bit hazy. So I would even think before even thinking about screening for resistance, we have to kind of really nail down what's going on. But yeah, it's mostly a capacity issue, because it would be great to have a plant, an ʻōhia that's resistant to leucohea, juliohea, uh, neofusicoccum, or any other fungi that are related to that kind of canker disease. But yeah, not at the moment. We're kind of starting with the big one first, and then we'll see what happens over time.
And follow up, Kirsten says, I completely understand. Thank you for the great work.
Thank you.
And quick question, one more quick question from me, and then we'll move on. As far as your clones and your seeds, I know that each of our islands have different varieties and even different species of ʻōhia. And I know that you did mention that you're looking at that. I'm wondering if you have um, representation from, say, all the islands, or if there's anything we can do to help make that happen.
We don't. We have some representation from different islands. The it, second to Hawaii Island would be Maui. Has been kind of sending seed to us, because we have an agreement, and they're kind of helping support our work through, by us screening some of their material. Really, again, it's a capacity issue. We know how big our greenhouse is. We know how many seed families we could support, and for growing and screening. We have 2 inoculation screening greenhouses, and we have 1 growing area here. So we kind of know how much we can support, and then we divvy it up um, between them. And really, for this first kind of round of seed family screening, we're focusing on working with land managers that actually have a lot of rod mortality and actively want to plant stuff out, because the last thing I want is to go through all this work, and then they say, oh cool, thanks, that's great to know, but we're not going to do anything with it. So we really want people that are ready to outplant, and so that's kind of where we are now. But the goal is to go to all the islands, and all the seed zones. So here at the map in the upper right corner, you can see there's kind of these little boxes here, delineations. Those are different seed zones, and so we do want to try to the ultimate goal is to have populations of ʻōhia that are locally adapted for their particular zone. Um, but that's the future, and right now we're really just working on kind of these rot affected areas. But yeah, we do have goals of expanding this and screening from other material of other islands. We've been in contact with DOFA and Kauai. All the islands have seed banks,
Right.
so it's
Okay.
there. It's all the capacity issue, basically.
Totally understand that. So when you're ready, let us know. We'll be happy to get you some seed from Kauai, as I'm sure other people here will be happy to share from other islands. I do have a question from Allison that came in. So can you definitively say that you've found some rod-resistant trees at this point?
I think I have a advisor that helps us with resistance screening. I think what we like to say is we're cautiously optimistic that we do. A lot of this stuff we want to rescreen because there are little mm, abnormalities or peculiarities sometimes with individual clones or trials. So I would say that we're finding strong evidence of variability in these populations, but and that we're cautiously optimistic that yeah, we have different levels of resistance. But at the moment, I wouldn't want to point my finger and say, "Oh, this clone right here, this is bulletproof, or this is completely resistant," without some follow up work. And that's normal. That's usually how this goes. You do the greenhouse work, you bring it to the field, you see how it goes long term, you put it in areas of fraud. Resistance breeding programs are decades long programs. So we're very much in the research pre-operational phase, I guess you could say. I'm conservative about kind of ringing that bell just yet, I guess you could say.
Understand. We do have a couple additional questions, but I'm going to save them from the end because I know you want to move on to the next part of your presentation. And these are some really good questions, though, so I hope everybody will hang around to hear those. And back to
Okay,
you.
I'll jump in. Yeah, and I might go a little bit faster through this because I know time is ticking. But I'm going to talk about a different project we have with Myrtle Rust and this Myrtle Rust Monitoring Network we're trying to develop. And this is in collaboration with two scientists from Colorado State University, Jane Stewart and Jorge Ibarra Caballero, both from Colorado State, both developing the technology and the assay that we're implementing here in Hawaii. So if you're not familiar with myrtle rust, this is what it looks like on myrtles, Myrtaceae, the family. It's kind of really known for this powdery yellow pustules that produce airborne spores. There's other rusts out there. There's coffee leaf rust, lots of different rust pathogens, but you know, if it's affecting a myrtle tree, then it's most likely myrtle rust, and that's how it got its name. Real quickly, it's Austropuccinia psidii, used to be called Puccinia psidii. You might hear it as guava rust, ʻōhia rust, but it's really bigger than that. It's the whole myrtle family across the world. It's an airborne fungus. It originated in Brazil, as you can see on the map. That's number one, so it's probably native to Brazil. Moved to Jamaica in the 70s, Florida. Oh, Jamaica in the 30s, Florida in the 70s, and it hit Hawaii in 2005. And since Hawaii, it's spread all across the east, really having a severe impact in New Zealand, Australia. It's really kind of sad how much devastation this disease is causing because their forests have a lot of Myrtaceae in it. Has a very large host range, 500 plus taxa. So that might be varieties and not just species, but maybe at a more fine tuned level. But yeah, over 500. And so it's a worldwide pathogen. In Hawaii, it was discovered in 2005 on Oahu in some nurseries. And if you remember rose apple, that was a host that was really hit super hard by myrtle rust. And here on Hawaii Island, at least, it kind of took all the rose apple out. There's still some remnant populations, but nowhere near the numbers that there used to be. And nobody really, I mean, that was an invasive exotic. So, um, I don't think it was really I don't want to say people didn't care, but it's like it was an invasive exotic. I'll just leave it at that. But you also have Eugenia coelauensis, which is a TNA species here, which is also heavily affected. So it definitely is affecting the native forest of Hawaii. And on ʻŌhia, it's considered moderately resistant, but there are these kind of waves of dieback that it can occur. And there was one that happened in 2017, which is really what brought us into looking at this again. There has been research in the past, but this is kind of what brought me into it, I guess you could say. So here's in the Kolau Mountains. Every tree that you see there that looks defoliated is an ʻŌhia that's got defoliated by myrtle rust. Here again, you could see. Good thing is this didn't necessarily kill all of these trees. Will Weaver went back a year later, and he has this ability to take these wide format photos, and he was showing that there is some regeneration that's occurring. They do have enough internal reserves to refoliate again, but some of the trees did die. And if this happens over and over, the trees won't have the energy to refoliate. So you can see how quickly things can get infected because of its airborne nature. Jane Stewart basically she sampled across the world fungi, the myrtle rust fungus, and basically looked at the genetics to see how different are they based off of what host they infect and like the population structure. So how similar are all the myrtle rust across the world? And really what it's showing is these three large groups. So you got the purple oval, the green oval, and the red oval. And basically they've kind of segregated into three groups. There's this Brazilian group that attacks guava and what's called Brazilian guava. There's this rose apple eucalyptus group in Brazil and Uruguay that attacks those hosts. So those are kind of clusters, and we call those biotypes. It's genetics with a host preference. And then we have what's called this pandemic biotype. So this is all the samples from Jamaica, Mexico, Costa Rica, Puerto Rico, Florida, and Hawaii. So everything that was captured in Hawaii is of this pandemic biotype. This is the only one that exists in Hawaii at the moment, which is a good thing in a way, because you don't want to have all these different biotypes here in Hawaii because, like I said, right now it's moderately resistant to the disease. So, you don't get waves of myrtle rust every year coming through and defoliating the forest. So it's kind of good that we have levels of resistance to myrtle rust. They did some bioclimatic modeling showing how, based off of the data they know of what environmental variables are needed to support myrtle rust, you could see that the pandemic biotype can basically infect every island, most parts of every island. If you go down to the lower one, that's C, you look at this rose apple eucalyptus biotype from Brazil and Uruguay, and it has a more restricted environmental range. But what's kind of worrying is in the past, many years ago, we sent ʻŌhia seed over to Brazil. They grew up the seed and they tested it against their local strains of rust they have there. And what they found is the strains they have, which are represented in this rose apple eucalyptus biotype, are way more virulent to ʻŌhia than the pandemic type we have here. So we kind of know that there are strains out there in the world that they can have the ability to do more damage to ʻŌhia than what we have right now. So this kind of paved the way for restrictions on importing Myrtaceae. So I'm going to kind of go through the different scenarios. So from international to mainland USA, to my knowledge, and I'll just say all this is to my knowledge, the best of my knowledge, international to mainland USA, there's no restriction. Mainland USA to Hawaii, it is restricted through this Hawaii Department of Agriculture ruling that says all plant material in the Myrtaceae cannot be brought in. Internationally to Hawaii, there is an APHIS rule. So APHIS covers international shipments that restricts what's called plants for planting, like plant material. When you think of potted plants, stuff like that. But when you look at cut flowers and greens, there is no restriction from importing internationally that material. Now, it should be said that it's not a huge, it's kind of a smaller window, but what we're looking to do is kind of see if we can close that biosecurity gap. And working with Rob Hoff from DOFA, Basically he said that, we have to basically establish this monitoring program. And the idea is that we can show that there's only one biotype in Hawaii and we want to exclude others from coming in. So that's really why we're doing this monitoring program. And so how we're monitoring is we're trying to use sentinel plants. So these are plants that are hosts for these different strains of the rust or biotypes. And they're kind of like living bait, living nets. And then we're hoping that if you monitor these specific plants, that if you find rust, you can send it to me and I could do this new DNA test that the Colorado State folks have developed. And the test will basically determine what biotype it is. And that's going to determine, have we had new introductions? Because other than that initial 2018 study, nobody is monitoring for rust at the moment, at least Myrtle Rust. So the workflow is that if you are a collaborator and we're reaching out to, land managers, people that are in conservation, that have the time and ability to do this monitoring work. But in regards to the sentinel plants, if you say, hey, Mark, I want to join, we can try to get plants to you. We'd probably source them from local nurseries in the state, get them into your hands. You would plant them in a pot or in the ground, monitor for rust. If you see rust, you will pack it up, and we have materials for that. You would bring it to the Hawaii Department of Agriculture for inspection, and they would either give you the sticker that you could ship to me directly, or you could just leave it with them and they'll ship it to me later on. We'll take those samples, we'll extract DNA from the rust, and we'll run it through this new assay that was developed at Colorado State to determine the biotype. This is how the test works. I'm sorry, I'm flying through this, but so let's say we have a panel of primers basically that will determine it's a yes or no. Is it pandemic or is it not? It's a color changing test. So it doesn't need like a full on lab to do this work. And then we're also helping Jane and Jorge kind of test their South African panel. So imagine you send me a sample and I extract DNA, and that is plant 21. So let's say I get DNA and I put a little bit of DNA in the pandemic panel at the top, and the same DNA in the South African panel at the bottom. Everything starts as red. That is the starting point. If there is DNA of the pandemic strain, the DNA will amplify, it changes the pH of the reaction, and it changes the color. So everything here you see at the top of the pandemic primer is, if it's yellow, it means it has successfully detected pandemic DNA. If it is red, it means it has not. So number 31 here is our negative control. This just has water. And this is just showing that, there's nothing wrong with the assay. It doesn't amplify anything because we haven't put DNA. The 32 is our positive control. So you can see that most of these are positives for pandemic, but number 23 here is not. And that's something that would raise kind of our red flag, and we would have to rerun the test. It could be that it's not pandemic DNA. Maybe we didn't get a good sample and we have to rerun it. Maybe somebody submitted something that wasn't even Myrtle Rust. So there's ways we can check that on the back end to make sure it is actually at least the pathogen, regardless of biotype. And as you could also, it kind of is the opposite. So let's say, look at number 25. If it's yellow for pandemic, it's going to be red for South African. So again, we're not really that concerned with South African, other than we're helping them develop the strains or the test. So for this work, we've been using like sentinel plants. Like this is one I have in my backyard. It's much bigger now, but the idea is, what we like about it is if it's close by and it's something you can keep your eyes on routinely. It's small. You can just pick the leaves. That's kind of the idea we're going for. But we also work with land managers and conservation people, that they just send us collections from the field. And that's totally fine too, because the more of a net we can cast, the better. The more samples, the better for this monitoring work. And if you want to join and you're interested, I have my email at the end. We'll send you a packet of just some guides of like, what does it look like, and tubes, and how to pack a sample. It's pretty straightforward. You kind of just get the leaf and you put it in the tube I give you put some of your information down, and you got to take it to the Hawaii Department of Ag, and they'll send it to me. I don't really have results yet because we're still kind of working through some tweaks, but that's the Myrtle Rust Monitoring Program. And yeah, if you refrigerate your samples if possible. And with that, yeah, I'd like to say thank you. If you're interested in the Myrtle Rust monitoring work, you can, or the ʻŌhiʻa disease resistance work, you can email me at my email there. akakaforest.org has a website dedicated to the ʻŌhiʻa disease resistance program, so you can get more information there. And I think that's it. That's my last slide. So thank you all for your attention and good questions. And I'm open to questions now.
Thanks, Mark. Great job making up time on that second half.
No problem.
Very impressive. We do have a couple questions I want to get to. But meanwhile, if Kylie's ready, I would love to put our post-webinar survey up, because otherwise I'm going to forget about it. Yeah, and if you want to stop sharing, that would be great too. And there it is. There's our poll. So if you would just take us only 2 questions on this one, couple seconds to answer it. Pretty quick, pretty easy. While people are doing that, I'm going to share a question regarding our earlier discussion on the ʻŌhiʻa disease resistance program from Jolene. Mark, she writes, I have a 400-acre ranch on the Kohala Mountain Road, and 20 years ago, I planted about 5 to 7 acres with ʻŌhiʻa. I raise cattle and sheep on the land. I do not have any noticeable problems with my trees. Would my forest be of interest to you at all? It is very isolated from people, but there are older original ʻŌhiʻa trees that were there when we purchased the property. So this is a land manager, what you were talking about earlier. That's kind of sounds like the group of people you're looking to reach and talk to. And you've just shared your email address. Would the best advice for Jolene be to reach out to you by email?
Yeah. I can't give you an answer off the top of my head. I have to think about it. But yeah, if you reach out to me, we can talk more about it.
Okay. And let me put your email again in the chat. It was mark.hughes@usda.gov.
Correct.
Okay. So Jolene, if you're still here, if you would reach out to Mark via email. And there was a question in the chat which seems to have disappeared. So maybe you answered it, but I think it's a good general question to ask anyway. And that is, it came in like from a concerned community person. Basically, what can we in the community do to help? And it was specifically related to Rod, but I also think, your second half of your presentation answered that question in another way. I mean, we're all concerned about saving ʻŌhiʻa, not just necessarily about Rod, right? So there are different things that threaten ʻŌhiʻa. So obviously maybe participating in the Myrtle resistance surveying program that you just shared is one way. And maybe are there other ways that you can think of specifically that you would share with a concerned community citizen about, what we can do?
Yeah, I think it's being kind of up to date on best management practices. So the University of Hawaii has their recommendations. The 5 things you can do. Of not moving material, trying not to wound trees. If you think you have a tree that has rod, try to get it tested. And then, if you are a land manager or you have somebody with an acreage and you're turned off to planting ʻŌhiʻa, I would say consider actually doing it. There's been a lot of research shown that you can replant in areas that have rod. The main thing is you want to avoid wounding agents. So weed whackers, ungulates, things like that is really what predisposes trees to getting infected. But and yeah, if you want to and just outreach, I think the more people know about it, the more people know he is important, how important it is, and critical to the watersheds. Um, it builds up some power. They can work up the ladder in regards to funding for work, because a lot of this work is always it's year by year piecemeal. Everybody's kind of fighting for or petitioning for cash from the government, the state. So I think the more support you could show, even as a community, that hey, we support this work, and maybe you can talk to your representatives to show that support, can help us downstream in regards to scaling up research and putting more effort into it.
Great insights. Very good points. Yeah, we always here on Kauai encourage people to continue planting ʻōhia, because here, and like some other islands, unlike Hawaii Island, where you guys have ʻōhia growing, in your backyards and in the KTA parking lots and things like that, we don't, right? So, even getting ʻōhia growing back where it once grew, is highly encouraged. And I just love the idea of increasing awareness, not only of ʻōhia's importance to our watershed, but to a whole host of our native flora and fauna. We talk so much, and this is the year of the forest bird. We talk about our forest birds, and so many of them are endangered, and many of them rely exclusively on ʻōhia. Plus, there's a whole host of invertebrates, from snails to insects and spiders, that rely on ʻōhia again. Some of these fauna we don't even know anything about, or even know that they exist. They're just kind of quietly surviving in the forest. So raising awareness for all of that, I think, is great. And while we've been chatting, a question has come in from Bill. Has anyone looked into the resistance/vulnerability to these diseases in the other species of Metrosideros in Hawaii?
The other species of Metrosideros. Okay, I'm trying to think. So, it's ongoing, I guess you could say. We are screening as we can get hold of seed, of these other species. And then I know Dr. Lisa Keith's lab in Agricultural Research Service here in Hilo. She's not necessarily in Hawaii, but she has looked at other Metrosideros species, particularly Metrosideros excelsa, which is a New Zealand species. So, through collaboration with New Zealand, obviously they have a large Metrosideros forest too. So they're very concerned. So there's been some work done on looking at levels of resistance in case it ever went there. And I think luckily, and a good thing is they found that it was fairly resistant. I mean, pretty resistant to the disease. I don't remember the details, but yeah, it was very optimistic of what they found for that particular species. So we won't know until we test, and we are, testing material of the other species and varieties as it comes, as we acquire it. I guess you could say we're working with the seed banks to get it. Sometimes there's not much of that seed available. It's remote, or it doesn't grow well in Hilo. So there's all these. It's never easy, I guess you could say. But yeah, we are trying to bridge that gap and figure out what else is susceptible in regards to Metrosideros on this island in particular.
Great. Oh, and of course, right when I think we're almost done, Gabriel, aloha. Would it be possible for me to share some of the information mentioned regarding rod and myrtle rust in this presentation with the community?
Sure. I mean, it's already out there. It's being streamed. So
Yeah.
yeah, that's the point.
Yep. Yeah, this is a public meeting, public webinar. And like I mentioned earlier, for those of you who may have missed it, this webinar is being live streamed in at least 2 places that I know of, one on the Rapid ʻŌhiʻa Death Facebook page, as well as on the Kauai Invasive Species Committee Facebook page. And yeah, I think we probably have covered it all. So we did pretty good on time. Thank you. Appreciate that, Mark. And I
Yes.
appreciate everybody else who's here, and like the group pretty much stayed for the whole time. So wonderful. And there are 3 more webinars that are part of HISAM, Hawaii Invasive Species Awareness Month. So if you're interested, check those out. And Mark, as always, thanks for all you're doing to help save ʻŌhia.
Thank you. I appreciate it. And, it's a big group. It's not just me. So a lot of people working on this
Absolutely.
together.
It
So
is. It is. But the research that you're doing is pretty fun and exciting. So we look forward to hearing more about it. And yeah, you're getting lots of mahalos in the chat. So
great.
check that
Thank
out.
you.
And yeah, thank you all for being here. Have a wonderful rest of your day and wonderful summer.
Great.
Aloha.
Aloha.